So now I would like to introduce you to Dr. Lynn Paul, who is a clinical psychologist. She has extensive research within the world of DCC. She's a member of the IRC5, the International Research Committee, and she's the founder president of NODCC, which is the equivalent of us in the US. So definitely a big expert in this field. Enjoy her talk.
Hi, I am Lynn Paul. I am a licensed clinical psychologist by training and a neuroscientist by vocation. That's what I do now. I work at Caltech, which is a very scientifically focused institution. But I trained with Dr. Warren Brown at Fuller Seminary, where I did a lot more clinical work. And I've been a part of this community starting 30 years ago when Dr. Brown and I started this particular project. We are part of an organization called the International Research Consortium for the Corpus Callosum and Cerebral Connectivity. IRC5 makes life easier. And our research group includes people from around the world who are studying corpus callosum disorders, various types, from the point of view of molecular development of the corpus callosum all the way to behavior. So it's a very unique group of scientists because we cover a lot of different fields of science, and we're all focused on this one broad topic, but coming from different cultures and different backgrounds, it's really important for us to have an opportunity to sit in the same room and talk with each other. It's a lot harder to overcome the accents and the terminology differences and the perspective differences over Zoom, even though it's great. So we had 2 days of scientific meetings the last couple days, and they were incredibly productive. Hopefully we'll have a paper out soon as a group. And I just wanted to thank Corpal. You provided some of our treats through the day, gave us coffee to keep going, lunch, and it was a nice help to us as a group. We have a little bit of funding that we've gotten from one source and kind of keeps us going as far as some computer services, but we are not a funded group. So it's figuring out how to resource ourselves to do this collaboration is a challenge. So I really appreciate Corpal's generosity in helping us have this meeting. This IRC5, just to give you an idea, I think these are most of the countries where we're currently located. Some of you participated a couple years ago. We did an online conference over multiple days, and we had people from 40 different countries participate in that, all continents except Antarctica. And it was just amazing to have the opportunity to have scientists and clinicians from around the world interact with each other, talk about what's the best way to diagnose this, what are the best practices right now, and share that with all of you. So I encourage you to keep your eyes open for future opportunities like that to participate. A lot of that information is also available to you online. Recorded all of those sessions, so you can get to them through the IRC5 website, irc5.org. So there's a lot of really great information. You're getting a little taste today, but I encourage you to go check that out. I'm going to give a lot, pull back. Joe was giving you an idea of what an individual researcher does focused on a particular area of study. I'm going to pull way back and give some broad strokes about this condition. All of you know what the corpus callosum is generally, and it is the largest single structure in the brain. It has over 200 million neurons or around that. One of the recent findings, one of our group discovered is that most of the connections actually connect parts of the brain. We used to think it always connected this part to this part that matched. A lot of it is not doing that. It's connecting different parts. It's a very new finding, very kind of reframing how we think about the corpus callosum. It develops in utero, so between the 10th and 15th 8-week gestation. Many of you probably got a diagnosis by ultrasound, and it usually can't be seen on ultrasound until after 20 weeks. Oops. A few background things to have in mind. All of the corpus callosum nerves that are going to be there are present at birth. So you're not going to get new ones. You can't add to them. But they do continue to develop. The corpus callosum develops through the course of your life, being that they get more efficient. So it says myelination is completed. That's the insulation that helps those nerve fibers function much more efficiently. Yes?
We can talk myelination completes in 2 years, and that's when we're having a repeat MRI. So is that?
Well, you get a period of development up through about 2 years, and you do have a lot of change. And the myelination that occurs to that point allows you to get a much clearer distinction between white matter and gray matter. So an MRI that you had prenatally is going to be harder to read, or even early on. But by 2 years, you get a really clear image. But that continues. It's not that the myelination is completely complete. You really have a period of time, early development, and then in girls, it tends to be 8 to 10, boys 9 to 11 or 12, where you get another round of development. I'll come back to that in a minute as part of our theory. You can see in these images how the corpus callosum grows over time. It's even at 8 months of age, it's all there. By 8 years, it's a much more fully developed, and that's what I mean by the contrast. It just becomes much clearer. Now, the last 2 days, especially all day on Thursday, we spent a remarkable amount of time talking about the specific terminology. And one of the things that's, I think, kind of great about agenesis of the corpus callosum is it is an anatomic diagnosis. You talk about autism, ADHD, those are behavioral diagnoses. And there are tons of people trying to understand what parts of the brain are involved in those behaviors. Well, we are starting with a behavior, or with an anatomic finding, and then looking for the behavioral consequences. And they merge. There are some, I would say there are people with agenesis of the corpus callosum, or CCD, comment on that in a minute, who have autism, meaning their behavior fits that description. Autism is not a single thing. I do a lot of screening and testing of people with autism, adults with autism, and there's a lot of variety. It's not a single condition. So it's a behavioral description. So if you get that behavioral description, that's a commonly known thing, and it'll help your child get services. And at this point, we think often those services probably are helpful. We don't have great data to support that. But I want to make it clear, sometimes people, professionals will come in and say, oh, well, we gave you an autism diagnosis or an ADHD diagnosis, but now we found out that they have ACC, so they don't have that. Which basically means we're not going to give you any services. Well, they're comparing apples and oranges. There is nothing in the diagnostic manuals that says if you have ACC, you cannot have a diagnosis of autism. One's behavioral, one's anatomic. And if you need to fight that, just get in touch. Done that fight many times. So things to know. The broad term is dysgenesis, meaning it's an unusual formation. It didn't form normally. That covers agenesis, meaning none, and or, which is on the left here, complete absence of the corpus callosum. Our quote-unquote normal brain is my beloved Warren Brown. We all use Warren's corpus callosum in our images. A partial agenesis means you can see in the orange, there's this little blob. It's part of the callosum is there, but the rest is missing. And we still call that agenesis because the entire length of it has not been formed. Hypoplasia is where you've got the full length of the callosum, but it's thin. Now, I will spare you all of the discussions that we had about short corpus callosum versus long corpus callosum versus thick corpus callosum. Any of you have questions about that, we'll come back to it. But I want you to know that those are the kind of things that we as scientists and as clinicians are trying to figure out. What are all these different types of dysgenesis? And is there a difference? Because to date, what we have been able to focus on is really broad scope of complete isolated absence versus everything else, okay? So our precision in prognosis is really poor still. And we're working on it, but I'll get into that more in a minute. It's more common than we thought. It's probably actually more like 1 in 2,000 live births. One of the things that Dr. Brown and I are both very pleased and proud about is that there's actually a diagnostic code now. There was not until 5 years ago that was specific to this condition. It's more common to have a corpus callosum malformation of some sort in developmentally disabled populations, and the causes are varied. And so I'll talk about this a little bit more in a minute, but the number of people that we really don't know what caused it is still very, very high. I think that's enough for there. So agenesis of the corpus callosum by itself, many of you have your reports and you're trying to make sense of what that says. It has a few anatomic features that seem to be related specifically to the corpus callosum development. They're not a separate thing. They're part of the fact that you're missing the corpus callosum. So one is, here's a normal corpus callosum. And the anterior commissure is this little bitty connection that's there in a typical brain. Often it's typical in a brain that doesn't have a corpus callosum. Well, if you compare the size of those two, and you say, this is all that's left, and that's got to do all the interhemispheric transfer, no wonder things are slow, right? You don't have that gigantic connection between the hemispheres, you just have this little tiny one in agenesis, right? Going back to the other anatomic features, colpocephaly, some of you may have heard this, is this enlargement of the posterior part of the ventricles. It's very characteristic specifically to agenesis of the corpus callosum. That's why some of your kids you were told they have hydrocephalus, or maybe on ultrasound, hydrocephalus was the concern. You see enlarged ventricles, that's more common, that's the presumption. We'll come back to this, but it didn't necessarily mean your child had hydrocephalus. It meant that they had colpocephaly, most likely. Another thing that is very specific to agenesis is if these fibers, so in brain development, you start with these two hemispheres, they kind of make a little bridge, and then those axons, the nerves that become the corpus callosum, cross. And then that little bridge disappears, and you have a corpus callosum. If there's no way for them to cross, they don't have a bridge, they will turn and create this bundle that goes from the front of the brain toward the back, called a Probst bundle. And it runs along front to back. What it does, we still don't know. It's active. We've got some studies that indicate that it is an active nerve passage, and it's presumably active because it didn't disappear. Is it adaptive? Is it helpful? We don't know. Does it cause problems? We don't know, but it's there. So if you got that, if anyone talks about Probst bundles, again, I wouldn't get sidetracked by that. It's just part of the picture. And as I said, right now we're still in understanding differences, very broad strokes, isolated, quote unquote, with colpocephaly, maybe a few other small malformations, versus ACC plus, meaning a corpus callosum malformation plus other brain malformations or other genetic malformations that have multiple implications. In the isolated group, the percent that have a genetic anomaly that we know of is still very small. So some do, but the majority don't. That's why sometimes we can't just say, if you have isolated agenesis, you're going to look like this, and all of the people with isolated agenesis do really well. Because some of them actually have have some different cause that's impacting the rest of the brain, presumably, okay? One thing to know about the ACC plus versus isolated is simply that plus, our presumption is all those other things that are going on are kind of swamping out the impact of the corpus callosum being missing. If you're blind, that's probably going to be more noticeable and have a bigger impact on your daily life, as an example. Same with if part of your brain is malformed and not developed. If you don't have a frontal lobe in the right hemisphere, that might end up having a bigger impact than the absence of corpus callosum. Does that make sense? So I think I'll skip over this. One of the things for you all as family members, I'm just going to throw this out. Many of you probably know this, but there are always families that are confused by all of this. Who do I go to, and what do they do, and why do I care? I get lots of questions about, do you know a neurologist that can do such and such and such? To be honest, once a neurologist has diagnosed the condition and said your child has this, unless they're having seizures, unless there's some kind of major medical thing happening, it's a static condition. It doesn't change. So they're not going to have a lot of information for you. Also, a neurologist has a very specific scope of what they're trying to understand. Is there a brain malformation? Are there neural system problems? They're looking for, have you ever had an evaluation and they squeeze my hand, and they're looking for which side is stronger? Well, that's not very precise, but it's informative. So they're looking for big patterns, not for subtle differences. That's also why a neurologist may say, there are people that just are fine without a corpus callosum. Because if your threshold is neurologically impaired, seizures, hospitalized, major developmental problems, everybody else is fine, right? So it's about the scale of what fine means. A geneticist is going to talk to you about any genetic findings, genetic counseling. We'll talk about that a little bit more in a minute. A neuropsychologist, by comparison to the neurologist, does much more fine-grained testing. And it's looking at comparison to age-specific expectations of abilities in language, in reading, in math, etc. So if you want to figure out how my child's doing compared to their peers, what their strengths and weaknesses are, what they need to learn in school, what are their assets that we can build on for the future, a neurologist isn't going to tell you much, most likely. A neuropsychologist would be where to go, because they can map out the subtleties. And then there are all these behavioral therapists who are critical. One of the questions that you had was about intervention. And we don't have studies yet that track intervention. We are just beginning to get data on how interventions are used. Our presumption is, well, I'll come back to that. Interventions are valuable, is the presumption. Yes?
Throughout the day, if there's any documents we can take to our pediatrician or occupational therapist, because My daughter's been signed off from OT. We're getting no support because they want to adopt a watch and wait. And I'm fighting so hard to get her support while she's young. And we can change it, but I keep getting a closed door. And we have our biannual review with our paediatrician this week, actually. So I'm just really curious on how I can help convince to unlock support rather than adopting a watch and wait because she's doing quote unquote well, even though there's lots of things. So that would be great.
You got it. I'll give you some language at least for that. Of course, Rhonda's booklet is a great resource, but I'll give you at least some language for this. Yeah. So,
sorry, can I get that as well?
I'm gonna.
I'll throw it over.
Yeah, it'll be in this talk. Yeah. So instead of going through a lot of our research data, I'm going to give some broad strokes information, but let me give you a little background. Dr. Brown has a great story about how this whole field of research started. He was doing research on interhemispheric transfer, so EEG, looking at brain waves in this side and how fast they move across the brain, and was looking at some of the people that had their corpus callosum cut. Some of you have learned about those split brain patients. And someone came along and said, hey, I've got this adolescent boy who's got agenesis, who's born without a corpus callosum. You want to study him? Sure, that's what scientists do. We had, like puzzles, we tackle them. Well, at some point he had 2 adolescent males who had this complete absence of the corpus callosum. And he took them out to lunch with their parents, or their parents went out to lunch. And the parents started talking. And there was your son does that? Our son does that. Does he do this? And he starts taking notes on all the things that he realizes, this is the story. Parents don't care about how quickly the electrical potentials cross the midline. The story is there are all these social deficits and there are these functional deficits. And so he, at that point, luckily I happened into graduate school and joined him, and our question was, and has been for the last 30 years, what is common among people who have who were born without a corpus callosum? What do you need the corpus callosum for? And that's different than what do you need it for, like the split brain studies, because this is what do you need it for even when the brain has done everything it possibly can through development to compensate for its absence. So our question, the reason we've mainly studied agenesis, so complete absence, and the higher functioning people without chromosomal abnormalities, is because, yes, we can test them, and they can do our complicated and interesting tests that we set up, but also because we wanted to know what part of this is due to the corpus callosum absence. And the best way to do that is to look at the people where presumably everything else is pretty much intact. So a lot of times families get frustrated because we haven't been including everybody. But we've been trying to answer this question that many of you ask is, what if this is because of the corpus callosum issue, and what if this is my kid? Or something else, right? So our focus has been on what's the corpus callosum's purpose. And we've tested all sorts of cognitive abilities, from very basic things, academic issues, language, everything you would cover in a neuropsych assessment, as well as empathy and theory of mind and social interactions, and try to map out what we think the syndrome is. And I'll come to that in a minute. Part of why I'm presenting this right now is because I want you to understand the terminology that I'm using. We have used agenesis, and I try to be very specific in this because there are times that I'm talking about what we know about agenesis. We know this because it's about people who are missing a corpus callosum entirely. We presume that it generalizes to people who are missing part of the corpus callosum. We have some data that shows that it does, or people who have thin corpus callosum. But If it's a study that has included everybody, then I will probably call it DCC, because that's how I'm used to calling it, dysgenesis of the corpus callosum, which is the same as corpus callosum dysgenesis. No difference. Same thing, different acronym, okay? And so when you see that, just know that I'm probably being specific about whether it was a broad study or more narrow. So some of the myths. First one I mentioned, large ventricles means there's hydrocephalus. If there's hydrocephalus, then it builds up pressure. You have all sorts of problems. Very few people that I have met with corpus callosum dysgenesis or agenesis have to have a shunt. There's not a pressure buildup. They just have those enlarged ventricles. The example I showed you is not a very big enlargement. There's some that it's huge. And I'll tell you anecdotally, I have not seen any clear pattern of the size of those lateral ventricles and ability. I can think immediately of a woman who graduated from college. It looks like she almost has no brain tissue in the back of her head. It's very thin. So there doesn't seem to be a clear association that impacts function. Another common myth or question that comes up is genetic. What does it mean if it's genetic? Does that mean that I passed it on or they can pass it on? Not necessarily, and I'm not even going to attempt or touch on the issue of genetics in detail, except to say that we have found a surprisingly large percentage of the people who have corpus callosum dysgenesis had a mutation. The ones that do have some kind of genetic mutation, it's a de novo mutation, which means it's not inherited. It happened to this little person themselves. Something changed in their DNA in one of the chromosomes, and it's specific to them. It was not passed down. Does that make sense? So when you hear de novo, it means out of nothing, kind of. It's a new mutation. Sometimes those can be passed on, sometimes they aren't. Sometimes they're passed, you can inherit a mutation and it'll cause a corpus callosum malformation, and sometimes the same mutation doesn't because of other factors. So it's not a simple question, basically. That's why you need to go to a genetic counselor, because they will not only be able to tell you about that gene, if they found one, but also about your particular circumstance and how perhaps the heritability factors involved, okay? And so it's complicated. There also, you may see studies that say the genetic impact or influences that cause corpus callosum malformations are very similar to the ones that cause autism or schizophrenia. Yeah, they overlap a lot. But genes aren't just on or off. There's also degrees of on and off. And there's multiple genes partially on that together have an impact. So that's part of why this is a complicated question. Complete versus partial, or hypoplasia. We tend to think that some is good, more is better. A lot of families think, oh no, but they have partial agenesis. That's a good thing, right? Well, our colleagues in Brazil did this study, and I think it's a great way of showing some of the complexity of what partial agenesis means. If you look at the midline pictures of these two, and you look at the little blob of corpus callosum in both of them, it looks like it's a pretty similar area, right? And sometimes reports that we get will say, they had the genu of the corpus callosum. And our presumption is, if it's that part of the corpus callosum, it's going to be doing what that part of the corpus callosum would do in a typical brain. But these tracts, these pretty colored pictures, are fancy technology that tracks where those fibers are going. And you can see this is coming from the middle, and the fibers that go out to one side. and this is looking from the top down, so looking at how they go out. That little blob of callosum is going to the frontal lobes and up to the middle portions of the brain, probably the sensory motor cortex. This little blob, same place, is all going to the frontal lobes. Look at it here, it's all frontal, and has this weird bundle that's going from the front on one side to the back on the other. It's not doing any of those connections to the sensory motor cortex. So, is partial always better? Is partial genu the same in every person? No. It's a really complicated picture. And so when we do behavioral studies, we might include people with partial agenesis and give a description and say how they compare. But that adds a lot of variability, and we can't be sure. So that's also, and the same thing goes for hypoplasia. How those connections are distributed can vary, and how the reasons why it's thin can vary. And so part of what we're doing as scientists is trying to get as many people in the world to give us their MRIs and their genetics and their behavioral data so we can start to figure out, are there patterns? Like Joe was saying, not just CCD versus non, but hypoplasia versus partial versus et cetera. And the only way to do that is to have large groups. Warren and I have focused on getting a large group of isolated, high-functioning adults with agenesis to come up with our syndrome. Next steps are this discerning the different variations. So some is good, more is better. Well, nah, maybe, nah, but we don't know. Sorry, I'm not giving a lot of good answers, am I? I'm a scientist, I look for problems, I guess. This is a really important one, and this is gonna start to get to your question. How many of you have heard somebody say, oh, I've met people that have a callosal agenesis and they're perfectly fine? Mm-hmm. Again, what's your definition of perfectly fine is my first question. And please have them enroll in our research so that we can have some example of this. But more importantly, all kind of jesting aside, you're talking about somebody whose brain is wired very differently than most people. There's a lot of variability in all of our brains. But if your brain is wired differently, it's going to function differently. As an example, somebody who can drive a car and has access to a car and has to get around London has an additional set of resources for getting around London than somebody who doesn't have a car and is physically challenged and can only use an electric chair. So not only could they have a challenge of trying to get into public transportation, but getting into public transportation is challenging when you're in a chair, right? Can they both get around London? Yeah. Is it the same? No. Can somebody in a chair function at a very high level? Yeah. Do they have to do it differently? Yeah. Do they have limits to how they do it? Do they need extra scaffolding? Yes. So we're going to talk about some of those factors. But that's one thing to keep in mind when they say, oh, your child's doing really well. Well, we know that my child is not doing it the normal way. So what does that mean? This is the syndrome that Warren and I have come up with so far. And what we were looking for are what are the things that we believe are caused by the absence of the corpus callosum. And one thing we know is if you don't have a corpus callosum, you're not as effective at getting information back and forth across the brain, right? So it's not as bad as if it was cut, but it's not as efficient. So limited interhemispheric transfer. The other thing that is very clear is processing speed is slower. Sticking with our car versus, now, anybody who's driven a car in London can say that's not necessarily the most efficient way to get around sometimes, right? It's not the fastest. But compared to the person in the chair, in the motorized chair in particular, it's probably faster, right? So the brain, without a corpus callosum, is having to share information, work together, do all of the things that a brain does in a less efficient way. Makes it slower. And that has a lot of implications. And when I say processing speed, it means listening to what I'm saying, it means following the teacher, it means adjusting to a new situation, all of that stuff that you were talking about of scanning, integrating information, inference, especially as you get into situations where more things go on. So our third component is the complexity issue. Even if you take out the issue of speed, when something becomes more complex, meaning it requires multiple types of thinking at the same time, or integration of multiple types of thinking, or if it's just has to be done under time constraints, some kind of complexity, the ability to do that declines. It does for all of us, right? So you think, well, yeah, you put me under time pressure and it's worse. When we compare somebody with agenesis to their peers, same age, the decline in the typical person and the decline in the agenesis person is different. Does that make sense? So that additional complexity in a task. Another great example of that is how many of your kids have had trouble learning to read? Right? If reading is a problem, how much history are they learning from reading that history assignment? You've just put two skills that they have to do together, and you're presuming that one isn't interfering with the other. But for that individual, it may be. So complexity is an issue. Now, out of these, we think that most of these are secondary deficits. We're not sure. It is possible, and we're going to be working with Joe and larger data sets to tackle the question, is inference, the ability to imagine and elaborate, is that a core problem, or is that secondary to these others? Time will tell. For your purposes, it probably doesn't matter that much. It's a problem, right? So that's the imagination, creativity in red is our current question. But it plays out in all of these ways. These are the symptoms that we have found that are common in people, even the high-functioning ones with agenesis of the corpus callosum. That learning can be slow. So often we'll hear they're learning delayed, or you might have a child who had a reading deficit, and then they don't anymore. Well, it was really they were delayed in doing it. Everything that's new is going to take them longer to learn and be harder to learn. Take more practice, take more repetition, etc. So just a global delay in learning something new. Non-literal language comprehension, so the ability to read emotions and inflections. When something, you start to hear a sentence and it turns a different direction at the end, that kind of thing. Flexibility and adjusting from what was planned to a new idea. So that looks a little bit on the autistic spectrum, right? And then all of these kind of social issues tend to come up. And so that's why you get a description that looks like autism spectrum disorder. It might look like ADHD. I think ADHD comes up because there's that time delay. If you're not always keeping up, I'm processing what you just said. You said 3 more sentences. I missed those 3, but now I'm going to pick up here. It looks like an attentional problem, but it's really kind of processing speed. And we have seen difficulties in basic things like recognizing emotions. Can you learn those things? A lot of them you can. How you learn them is different. So in this behavioral syndrome, a couple things that are key in how we understand it. As something gets more complicated, like I was saying, the ability falls down. So the blue line is average for somebody who's your matched age, gender, whatever the important criteria are. And the orange is kind of a global example of what corpus callosum dysgenesis or agenesis might look like. So complexity plays a role. Processing speed, same thing. The more faster something has to be done, you see this decline in ability. But that's also why you see inconsistency. I can do it sometimes, and I can't sometimes. I'm sure there are moments where you think, I know they can do this, right? You could do this yesterday. Why can't you do this today? Well, are there other things going on? Are there other distractions? Are their resources limited? So all of us have mid-afternoon slump, and we have ways of coping. This is kind of my guesstimation of what that might look like for somebody with agenesis, corpus callosum, or dysgenesis. There's a more rapid decline. Maybe there's recovery with a break. And then look at the slope. If you talk with people who have ACC, the adults will say, I'm okay, and then I'm not okay. I've seen kids doing some task with me, and they're doing fine, and then all of a sudden their head's on the desk. It's like, what just happened? They got overwhelmed, they got exhausted, meltdowns, screaming fits, coming home at the end of the day, and they're completely a mess. If you push them right then, do you find, oh, they've got reserves and they can keep going, or are they just more of a mess? Probably more of a mess, because you've got to get them some recovery to keep going. What I work with a lot with the adults with ACC or DCC on is knowing that this is their resource system. They're already working on backup in their brain. They've got to know, I'm getting toward the end, I can't do this if I don't get enough sleep, if I don't eat well, all of those things, they're not going to have the resilience that someone else has. That's why maybe you don't do as many after-school activities. Maybe you make those kind of decisions. Stress load, work, etc. Another thing, if we don't tell them, they won't know, they won't have reasons to not do things. If we tell them, it'll be an excuse so they don't have to do stuff. If we don't tell them, treat him like he's normal, act like he's normal. No. This is my favorite. It's not an excuse, it's an explanation. It explains why you may need to do it differently, why you might have to work harder, why we have these workarounds, why you have a tutor, et cetera, et cetera. Why we give you so much scaffolding is because of this. But it doesn't mean you get out of doing it. You still have to function in the world, right? As to the question of intervention and recovery, and this goes to your scaffolding question and the question that somebody posed to Joe, I have not seen a lot of just kind of automatic learning. I mean, we all learn from social pressure, right? And you learn what to wear, how to act, who's the bully, who to avoid, all of those things. You have a bad experience, you adjust your behavior. Individuals with DCC don't tend to adjust their behavior very well and may miss it entirely. So a lot of examples I hear are families saying, you know, the mean kid at school, they come home, they're crying, the next day they say it's their best friend. And this has happened over and over all year long. It's like they can't learn that this person is mean to them. So to presume that they're going to learn from those normal social pressures is a false assumption. You have to be much more explicit, much more detailed. You can't even necessarily assume that they'll generalize from one situation to the next without discussing the idea that it needs to generalize. which means for you as parents of younger children, talking through social situations, learning to think about them differently, breaking them down into details, that's the scaffolding we're talking about. Giving them tools to approach situations. And that has to start young. So to your question about early intervention, the more something is routine and automated in our brain, the less energy it takes, the less attention it takes. So whatever your child is learning now, you want them to drill it in, learn it, get it as solidly as possible ingrained in their system, however their brain does it. I don't care. People will also ask, do I need to do certain interventions where I cross? We don't know how those brains are adjusting. The brain finds a way to do what it needs to do. Put the next task in front of the child, work at it, repeat, emphasize, help them learn to do it so that they don't have to think about it as much anymore. And anything that you can't automate like that, use outside systems. If I put my keys on the little table by the front door, I don't spend hours every day looking for my keys when it's time to leave somewhere. If I use notepads and lists, it's my external brain on this piece of paper, or spreadsheets. Now, it may seem silly to tell a kindergartner, I want you to use a daytime planner. But if you have a kindergartner carry a planner, and they get used to having it, and this is where the teacher always writes down their assignments, even when they can't read, you've set up a system that will last for years. And you don't have to introduce a new system when they're an adolescent and don't want to do what you want them to do. So thinking ahead of what are systems that will last a long time that I might use now as an adult, but would be helpful for somebody who's cognitively going to need scaffolding. So that's why you go back and say, no, my child's brain is learning things differently. They need to learn it optimally so that any component of it is, they have the mental ability and the resources to focus on. So as I wrap up, it's not an excuse, it's an explanation, it's a reality. I think treating it as such is really the healthiest way, the whole neurodiversity, acknowledging we're not all the same. And that's just the way it is. The impacts change over time. One thing I didn't put here is that developmentally, one of the things we see is a child may be progressing pretty well. And what we often see is early elementary or mid-elementary school, they start to have more trouble. And part of that, we believe, theoretically, is that the corpus callosum and all those kids that have a corpus callosum is going through that next developmental phase. And these kids, so that would be 8 to 12, kind of that period. And things socially get more complicated. That's when kids start to have less explicit, like, I hate you, but more subtle indications that you're not the cool kid. All of those subtleties, all of those social things, theory of mind, thinking about what somebody else thinks, these kids struggle with that. So they don't lose abilities, but they fall behind where everybody else is going. So that's why we call it an emergent deficit. It may be that their problems become more apparent with age. Tackle one thing at a time. Really focus on it and make it automated, use routines. Be direct, be explicit. Unfortunately, you may have to have conversations with your individual who has DCC that you never wanted to have. One of the moms of an adult says, I know more about my son's sex life than I ever imagined I would have to know. But he's 40, and he still calls her about everything. And she's like, I'd rather him tell me what's going on than him get arrested for doing something that he didn't realize was not appropriate. So I give that as an extreme example of better to be explicit, be clear. last thing I want to say as I wrap up is we have, right now, what I'm doing in our lab is focusing on the broader description of the profile. So Warren is continuing to do work on adults. And Warren, would you just stand up and say hello? Warren Brown. Throughout the day, please talk to him. He's done a lot of work on the social process and cognitive issues defining this condition. And we're continuing to recruit adults for online studies and eventually in person. But the bigger focus in my lab right now is on development. So we are starting from birth to up to age 18 and following longitudinally to see what can we expect in those early years. So all of you who said, what's going to happen? And they said, we don't know. Well, hopefully we'll have at least some map of what happens. So this is an ongoing study. We're looking at how the kids with agenesis compare to kids at risk of developing autism because they have a sibling with autism, how they compare to neurotypical kids, how they compare to kids with Down syndrome and Fragile X and other. So we've got collaborators doing all of the same studies in different conditions so we can answer some of those questions. And briefly, for these studies, typically what happens is you enroll online. Jasmine, who many of you have talked with, would talk with you, make sure that you're up for doing this. The infant study is the first 3 years, but anybody can join now and do questionnaires online once a year and give us an update so that we can start to get a better picture of that developmental process. And lastly, there are resources. I think there's some ACCME books over there. Emmy and Me is about sibling relationships. If any of you want to talk about family dynamics, I'm happy to talk with you later in the day. There are lots of complicated components to that. There are also versions in Kindle form or ebook form in other languages, if that would be helpful. And then acknowledging collaborators. The infant and child study I do in collaboration with Jed Elison at the University of Minnesota. For people that are in the infant study, if you're in America or if you reach out to Jasmine and we try to figure it out, you might be able to come for direct assessment. So we're trying to get more feedback directly to parents and actually interact with your children. And we're comparing to existent data on autism subjects. Collaborators also here in the UK, Glenna Andrews has been here, Warren, and then Elliot Shear. And part of my funding is from NIH, the National Institute of Health in the US. But we're always looking for more. And that's not because I'm pitching that, oh, please get us wealthy, but funding for the IRC5 and funding so that researchers can come together so that we can share data with each other and have the infrastructure to do that will move us forward in this field so much more quickly. So there's a great potential to really revolutionize the field with substantial contributions, but not, Gates Foundation endowments. So there's room to really participate, as well as participating in research. So please make sure you reach out to Jasmine if you have an infant or child, Warren for adults, me for whatever the questions you have. Thank you for your time, and it's lovely to see you all.
Thank you, Lynn. That was actually an excellent talk. Excellent points, especially the, giving a lot of emphasis on the scaffolding. I'm a great believer of that. And for those of you that don't know, I've got 3 kids. My middle one, Jimmy, that Nicole knows very well, is my little one with, he's 5 years old, with DCC, his complete absence. But we do a lot of that at home, as you say. It isn't just enough to wait from school to do things. We do a lot of things, even during the summer holidays.
Yes.
And it's just all about doing as much as you can with your children in your own time, not just relying on services or school. So that's excellent talk, emphasizing this. maybe an example in terms of the genetics. I don't know if that will help understand people things a little bit better. When I talk to you parents, when we talk on the phone and all of that, helpline calls and everything, what I like to say is, because people have this question about genetics and the complexity of it. And it's very difficult to explain, and it's not my field of expertise at all. But what I say, I give the example of the patients I'm deal with, which is the cancer patients. A cancer patient ask a lot, is this genetic? Yeah, of course it is, because some mistake must have happened at some point. Is it inherited? Not always. So some protein, some substrate just didn't go right in that moment in time. And then perhaps that is what explains the picture that we have with our children, but not always is passed from the parents or relevant to the family genetic tree. I have one question.
Well, can I just say on that,
Yeah.
we're having a lot of discussions about this amongst the group. I'm not a geneticist, so I was asking them lots of questions about it. There is some information that genetic testing can provide that's not just about trying to understand causes, but there are some genetic markers that indicate other systems may be involved. So it might be a flag that there are other things to be watching for. There are elements that would indicate that it is heritable, which you would want to know, presumably. But often, and so in utero, they might want to do genetic testing to see if any of that is available to inform. But often, for your day-to-day life, apart from those things, it's not probably going to impact you dramatically. But I'm not saying don't do it. It's still very useful for us in the future and all the other families.
I think also it's about how do you interpret genetic tests? Because you might find, well, there's chromosome 1 and whatever mutation. Is that relevant? Or are you getting hooked up in a information that you just drag on for the rest of your life? I have a question. So there's these individuals that have the famous cysts, the interhemispheric cysts. How does that come into the picture in the context of ACC? We spoke about Probst bundles. Maybe it's a good thing, we don't know. But then how does the cyst comes into this?
It's a great question. We don't know as far as outcome, because I've seen people with interhemispheric cysts, big ones, that do fairly well. It depends on the cause of the cyst, when it developed. One of the things to know about the corpus callosum is what we think is outcomes are going to depend on the cause, meaning the mechanism. So did something disrupt the way the nerves travel and where they go? Or did something disrupt the formation of those nerves in the first place? And if something interrupted the travel of the nerves across that midline, was it a protein? Was it information from the gene telling the nerve where to go? Or was it an obstruction? So you can have a cyst, a lipoma, something that physically prevented the callosum from crossing. And that's a very different picture. So those kind of factors would make a difference. The cyst, as far as just the space in there, it's going to, we don't have specifics on that.
Yeah, thank you. Any questions?
Great talk. In one of your slides, you mentioned that 30 to 40% autism related.
Mm-hmm.
Yeah, in this one, is it 30 to 40% misdiagnosed as autistic individuals, or is it 30 to 40% with comorbid symptoms?
30 to 40% that we have found that have those symptoms. So in an adult sample, we did ADOS, very in-depth diagnostic observation, and we came up 20% or 26% of our group met criteria for an autism diagnosis based on their current behavior. Screening measures in younger kids have been anywhere from 30 to 45%. Dr. Scheer did a big study. So that's screening for those behavioral symptoms, not saying, boom, they have this diagnosis. I'm always hesitant to say misdiagnosed. I try to avoid that because if it describes the behavior, then that's what autism is. It's a behavioral syndrome. So,
And
So I was going to say on that, I think, like you said, when you get that diagnosis, especially in the UK, they just sort of group it as that. And then a lot of us, if you want that sort of ASD diagnosis, then you have to go private for that because they pull it up. They don't know enough about the condition, so they say that's what it is. And then if you need that, then you need to go. Normally, have to go private in the UK to get that.
so if you have an ASD diagnosis, you don't get an
So
autism.
so I think because they've got ACC and they see like the brain malfunction, they group it as the umbrella of that. They don't know enough about it, so they might say, "Oh, you might have traits of that." But then to get an actual diagnosis is quite hard. Like, so there's probably more on your numbers than that don't actually get an official diagnosis, I think.
Right. So our numbers are based on actual testing with the participants, not based on how they're diagnosed outside. But you're right. They're probably, if we surveyed how many of you have gotten an autism diagnosis, it's probably less than that because people will discount it.
Excuse me, hi.
Hi.
Can you please touch upon neuroplasticity? And as you mentioned, myelination goes on until 8 to 12 years of age.
Mm-hmm.
Is it in any way related to neuroplasticity and whether that helps in development more?
So neuroplasticity, in a broad sense, is happening for all of us all the time. Right now, your brains are learning new information, and so it's reinforcing connections between nerves that are learning, that are encoding new information. It's not necessarily plasticity, like making big dramatic changes in the architecture. So the neuroplasticity that's so important for ACC is the brain coming up with ways to function better.
Alternate pathways.
We call it alternate pathways. I would say it's alternate systems. There's some evidence that there is a recruitment of pathways, like the anterior commissure may get used for a lot more, or in some people you may have an enlargement of the anterior commissure or posterior commissures, and that would be true plasticity of the neural system, like architecture that happened before birth. Or it may just be recruitment of those systems, like we just use it for anything we can. It's not bigger, it's not more efficient, it's just used a lot. Does that make sense?
So are the regular interventions that we do, as you said, early interventions, do they kind of help with development of new pathways or?
Yes. Well, they are reinforcing whatever pathways that brain has come up with. So, yes, that is what learning is. Learning is capitalizing on neuroplasticity. And if the brain doesn't have something, I mean, one of the other things we study is people who had a whole hemisphere removed surgically. And one of the things we found there is the brain kind of recruits neighboring areas of the neurons to help do whatever task it's doing. So yes, there's plasticity in the sense that functionally it gathers assistance, not necessarily like it's building new paths that you could see. Does that make sense?
Is it in any way related to developing seizures later on in life, or how many children with agenesis ACC develop seizures later in life?
I don't know the incidence of how many develop it. There is a higher vulnerability in agenesis broadly to developing seizures, and I've heard incidents, there may be some more vulnerability. There are things that can trigger a seizure for many of us: fatigue, heat stroke, certain types of stressors. You might not have epilepsy, but you could have a seizure. And so somebody with ACC may be more vulnerable to those kind of events, but not necessarily epilepsy. But I won't speak to the development of epilepsy. I'm not a neurologist.
Okay, thank you.
You're welcome.
Got a question.
I think on one of
of your earlier slides, it said about adults sort of having incidental findings later in life. And then you had around the percentage of the occurrence in the natural population. I just wondered, at what year or timescale did they start looking for like corpus callosum in prenatal scans?
They'll start looking at 20 weeks.
Oh, sorry, I probably wasn't clear enough. So was it in the 80s or the 90s? Or because
right.
yeah, until my son's got ACC, but obviously it was something I'd never heard about. But I just wonder when they started looking for it prenatally.
The earliest, I don't know totally empirically, my experience of it was there were cases people that I met that were diagnosed in the 90s in utero. It had to do with the quality of ultrasound. I think the rate of diagnosis frequency that we've just experienced is because of the frequency of using MRI now. And Warren and I would tell you that in our experience, when we first started doing this, it was all just people that got diagnosed because they had a car accident or something else. Gradually, it got to where there were more that were diagnosed as children because there was a parent who kept throwing fits and was the squeaky wheel, or the kid looked like he might have epilepsy, so we got more child. And now it's predominantly prenatal diagnosis. So it's really shifted dramatically in the last 25 years.
So great question. So Dr. Toma Ricci is coming in the afternoon from the Evelina Hospital. So he will be fantastic to answer that question as well. I mean, yes, the fetal MRIs have been happening in the last decade, no more than that. And in the UK, so it's a different system than in the US. So here we've got the midwives, as you know, that are taking you through the process of your pregnancy, and they deliver the babies here. And they do the scans as well. So you don't really see the obstetrician here in this country unless there is a medical reason. So it's only, I would say, in the last maybe 8 years in this country that the scanning skills of the midwives has improved. And if you were to go a decade or more ago, the scanning used to stop at the neck. They weren't really scanning beyond to see what's going on in the brain. It was just, their expertise and what they were trained to do was kind of at that point. And it's in the last less than a decade, let's say, that ultrasound and ultrasonographer scanning has come in, and you don't need to be a physician to do that. And the midwives have trained, and now they're scanning above the neck. So that will increase the pickup rate and all of that. So I would say definitely in the last decade, the quality of the images, the fetal MRI is present, the quality of the ultrasound is much improved compared to before. So you are able to pick up an abnormality as early as 20 weeks, and obviously clarify as you progress through the pregnancy. But you can ask that question to Dr. Ricci when he comes in the afternoon, okay? Thank you.
Thank you all for your attention. Appreciate it.